Good afternoon viewers and welcome to the EduSat lecture in the science stream. Today we have with us Dr. Eklavya Chauhan from the Department of Botany, Deshbandhu College, University of Delhi. Today he will be talking to us about epistasis. Epistasis is interaction of alleles of genes among themselves. As we all know, we have millions of genes and not all of them are working independently. The genes, their alleles, and more importantly the gene products interact with each other. They may suppress the expression of one gene, they may upregulate the expression, or may give you entirely new phenotypes. So what is this epistasis and how can we determine this? How can we view it by doing simple crosses? This we will be studying with Dr. Eklavya Chauhan. So without much ado, I welcome Dr. Eklavya Chauhan to our studio.
Thank you, Unnati, and thank you very much for a very apt and to the point introduction. What we have discussed in our earlier two episodes of non-Mendelian genetics was that how did we depart from the expected ratio of 3:1 or 9:3:1. Today our emphasis would be that all the two parts that we had discussed took a basic premise that the alleles are working among themselves. A dominant allele is dominant over its recessive allele working at the same locus, and it has nothing to do with the other genes or the other alleles which are located on the other loci. So, the idea today is to move a little away from the monastery of Mendel, of course, underlying the fact that all the principles of Mendel are still universal, and in the words of William Bateson, the person who had given the term genetics, when he had written a book in 1909 entitled the Mendelian Principles, he said that everything that Mendel has said is universal. Still, we must go beyond his monastery and see whether there are any exceptions. So, for the past two episodes, we have been discussing these exceptions only, and today let us see, as Unnati very rightly pointed out, that genes, are they always working all by themselves, or are they even interacting or even trying to influence the effect of other genes, no matter whether they are at the same locus or another one. So, that is the purpose for today's talk. If one looks at a normal dihybrid cross, the basic idea would be that the two parents which are true breeding, say for example, the classical experiments of the garden peas of Mendel, then basically we would have in the F2 the ratio which is the phenotypic ratio of 9:3:3:1. This would also constitute the first law, and that is the law of segregation, along with of course the second law, which is the law of independent assortment, or the so called, more popularly called, the principle of random hybridization. So, this very clearly points out that a particular character has nothing to do with the other character, and they are always going to segregate independent of each other. So, if we were to take seed shape and seed color, the final ratio, the phenotypic ratio would be 9:3:3:1, and we could empirically derive the fact that the genotypic ratio would remain the same in all the cases, but then it would depend on the interaction between the genes and their alleles, and of course, never to forget their interaction with the environment. So, that would be giving us the final phenotype. But in a normal classical genetics, everything is so goody and everything is so independent of each other that the ratios are absolutely explicit, as is seen now in the form of the seed shape and the seed color phenotypes, and there is absolutely no confusion. The first idea actually came to Bateson and Punnett, the famous duo who gave the laws a new dimension after Mendelism. They were the first people to have discovered that one can go beyond the expected ratios of Mendel, and if not, the interpretations could be different, as is the case in supplementary, as the word suggests. There is some supplementation, one helping the other or modifying. It gives us perhaps a false idea that perhaps the ratio is modified. We will see at the end the ratio remains the same, but things are so very different. They were the people who observed that in different types of chickens or poultry, there were different races, and they could be recognized on the basis of their combs. We would have seen umpteen of chickens, but we never bother to look at their crowns, and that was the type of combs that they possess. They found that there was a Wyandottes type of variety which had a rose shaped comb on their head. There was still another one which were called as the Brahmas. They had a pea shaped, like a grain of pea, the smallest one. And the most common which we see even by the roadside, the cocks or the male chickens which have a single type. So, they were fascinated as to how there are different types of chicken having a different morphology of their combs. Now, when they crossed a true breeding Wyandotte and a true breeding pea, the genetics came later, they were surprised that the product in the offspring was a totally different phenotype. You can see in the picture that it was a new breed altogether, which was the Malay breed, and the comb was something like a walnut, which was quite a ruminate one. Now, this was very, very interesting. In fact, it constituted the first evidence that a trait can be influenced by more than one gene, because right now we are just talking about one gene which is controlling the seed shape or the seed color. So, that means they were the first to have suggested that actually the two genes are independently assorting, no doubt, but then both of them are influencing the same character. Now, this particular idea was hitherto unknown, because Mendel never mentioned this. He said seed shape and seed color. So, that means they are two different things. Here we are talking about just one character, and that is the comb shape. And then they gave the different types of notations of the genes, and they said that it is R and P which are the main genes. The capital R would be for rose, and it would dominate over small r allele, which is for single. Likewise, another independently assorting, when we say independently assorting, naturally R and P would occupy different loci on a chromosome, and the capital P would be for P, and it will also dominate for single. But the moment R and P come together in a cross, they would simply modify the phenotype and it would become a walnut. So, this obviously was a very fascinating idea, which gave us the clue that if we have a single combed chicken, it must have small r, small p, that means a double homozygous recessive alleles. In no other case we could get a chicken with a single comb, and wherever we have capital P and no capital R, the shape would be P shaped, and likewise the capital R with no capital P would amount to a rose morphology, and the moment R and P would be together, they would somehow interact, they would somehow help themselves in such a manner that the entire morphology would get modified into a walnut one. Now, we find a pictorial depiction of the different types of the progeny in a cross, where we find that in the checkerboard of the F2, where we have the different types of permutations, mind you, the genotypic ratio has to be 1:2:1:2:4:2:1, which is the typical genotypic ratio for a dihybrid. But then the whole difference lies in the interpretation of the phenotypes. Still confusing, the phenotype is again 9:3:3:1. So, that means how does one get out of this confusion? The idea is that this is a case of an interaction, because these two independent assorting genes, no doubt they are there, but they are affecting the same trait. In this case, it is the comb shape, and we would find that there are different combinations, as I just said, with P and R, which are interacting with each other, and that means this interaction has to be at the biochemical or at the cellular level. So, that means somehow the morphology is changed by modifying the basic character. The basic character is comb shape, and the mere presence of two genes has modified it in such a manner that an entirely novel, an entirely new morphology has come to fore. So, that means this is some sort of collaboration between the two genes, some type of supplementation, and hence the term supplementary or collaborator. These are different terms used in vogue, but then the old classical literature always talks in terms of modifying genes. Just to summarize this whole idea is that the genes which are present on different loci, because had capital A acted on small a, we would not have been excited at all, because that's the rule. It has to dominate over the recessive allele, but then giving rise to a totally new phenotype, that means the how do we distinguish this from a normal classical dihybrid cross? One should always remember that in a Mendelian dihybrid cross, there are two sets of characters. For example, as we saw in the beginning, the seed shape and seed color. In this case, and there were 4 phenotypes which were segregated in 9331, but in this case we have only one set of character, and this one set of character is just comb shape, and it gives rise to 4 phenotypes. So, there lies the basic difference of modification. Is still very interesting and less discussed aspect of the modifying genes is inheritance of skin coloration in corn snakes. Incidentally, the corn snakes are not that poisonous. They are pretty cool in the fields. Their color is so very intriguing and so very picturesque that lots and lots of motifs on fabrics have been copied. As one can see, the natural color is a very beautiful repeating black and orange, and this color actually helps this reptile to camouflage with its surroundings and not get noticed and get killed unnecessarily. Now, that means it must be synthesizing both the type of pigments, and when both the type of pigments are there, naturally it comes to a mind that the gene also has to be more than one. So, it has been found later that the gene 1, that is the first gene O, let us call it, is controlling the synthesis of orange pigment, whereas the gene B is controlling the black pigment. Fine. If we have the dominant allele, then we have the orange one. If we have the recessive allele, which is denoted as O minus, then obviously the pigment would not be there, and likewise the B plus for black and B minus for the absence of black pigment. Now, where does the interaction come? If we have a genotype which is heterozygous, that means it is O plus and say any other thing for that matter, it could even be homozygous O plus, then there is and along with it we have 1 B plus. That means this snake would have a natural pattern because it would have both orange as well as black. If we have a situation of O minus along with B plus, so that means now both A and B plus are not there. So, that we would have one of the patterns, either there will be no orange if there is O minus, and there will be no black if there is no B plus. So, it is a very logical thing to assume. How would be the situation which we would envisage if none of the positives is not there? So, that means in the last case the O minus and B minus combination would give rise to an albino. This particular snake would have neither orange nor a black pigment. So, within this snake color, we have found that there are 4 different types of phenotypes, and a very beautiful cross summarizes the whole idea. If there is a female which is orange, that means if it is orange, that means B is not there in its dominant form, it is B minus, and if it is mated with a male, that means which is B plus, but O minus, then we would have in the first filial generation all the progeny which are camouflaged, and then an inbreeding of the camouflage snakes in the F2 generation would again give us 9:3:3:1, which, as I said in the beginning, confuses very well with the normal expectation of the dihybrid Mendelian cross. But then in this case there were 4 phenotypes to 1 character as against the 4 phenotypes to 2 characters. This brings us to another or the main topic of today, which is epistasis. The modifying genes was also an interaction, but somehow it failed to modify the ratio. It modified the empirical balance, all right, but then the ratio was not altered. Now, if capital A dominates over small a, fine, and so does capital B on small b, but then because they are at the same loci on a pair of homologous chromosomes. But then what would happen if capital A left its old function and started interfering with something different, which means, say for example, with a gene C or a gene B, which is not on the same locus at all. Now, this is reaching too much, and this would lead to lots and lots of complications, lots and lots of different types of unheard non-allelic interactions. So, we would find that gene interaction of this type would lead to a discontinuous variation. That means this may sometimes happen and sometimes it may not. So, the Greek word the epistasis has a Greek root which means stopping of something and literally to stand upon. That means if you really stand upon something, you are masking the expression or the movement of that particular object, in this case an allele. So, that means when the expression of one gene or an allele masks the expression of another, this is the phenomenon of epistasis. And of course, one should never forget that it is a non-allelic interaction between the alleles. As I said before so many times, it does not excite us at all. So, that means these non-allelic pairs would now be acting in which manner? It could be anything to our imagination. If this type of interaction may lead to, say, enhancement of a function, it would be called as, say, cooperative, complementary. Sometimes one of the functions may be stopped entirely. In that case, it would be antagonistic. So, that means the interactions could be positive as well as negative as far as the interactions are concerned. However, one would agree that there is some form of modification. So, the gene which stops the expression would be called as epistatic, and the one which gets modified or which gets masked is the hypostatic. In we will not go into the Merriam-Webster connotation, but the language of genetics always says that A is epistatic to B. We never say A is epistatic on, which literally means standing upon. So, A is epistatic to B, and if there are different types of epistasis. Now, the question is which gene is epistatic to which one? If a dominant allele is epistatic to a dominant or a recessive one, it would be called as dominant epistasis, as we could see, say, homozygous dominant A or a heterozygous A being modifying, say, capital B, or if sometimes it happens that A, that is a small allele or the recessive one, is epistatic to even a dominant non-allele. That means this is a case of recessive epistasis. I would modify my statement, not sometimes, actually it is the recessive epistasis which is the most prevalent one, and it is the most common one. And naturally, if this is going to happen, the 9:3:1 ratio is bound to be modified, and we would have different types of interactions. Before going into the specific examples, let me first ask myself a question as to why do the genes interact at all in the first place. What is the need? wasn't life good when A was just having its role with small a and it had nothing to do with capital B or small b. Now, we should look at the metabolism in a holistic manner. Wherever you have metabolic pathways, these are all stepwise processes. We talk in terms of a metabolic pool where different precursors are connected. One of the substance is acting as a raw material for the other product, and the other intermediate product could be the final product as a result of some interaction. So, that means everything is connected to everything, and it would therefore mean that whatever product we are talking about may not be a product of a single gene. It could be a product of a combined action of many genes altogether. So, if something happens to a combination of such genes, the entire machinery could be having topsy-turvy. So, that means if there is a stepwise conversion of the precursors, you can block the synthesis at any stage, and we have different types of combinations. Just to depict pictorially, we have a gene A, which is capable of converting a precursor into an intermediate. What would happen if an inhibitor was to be there and it would not allow the gene A to work at all? If it does not, then the whole story stops here only, because the whole chain stops, and then the gene B is useless. Sometimes it happens that the gene A is working, gene B somehow doesn't work, because there is a gene C which is stopping the action of gene B. So, that means in any case or in either case, the end product is not going to be formed at all. One should always remember, as I have said in the past, that dominance is an intra-allelic interaction where capital A allele is influencing small a, its allelic gene at the same locus, whereas epistasis is basically an interallelic interaction. That means, the capital A or the small a is influencing another non-allelic gene, which is capital B and small b. So, there would be an interaction among themselves, that is between capital A and small a, but then the main morphology or the phenotypic modification exercise would be in terms of A and B. So, we have said before also that epistasis and dominance should never be taken as synonyms, because the basic difference is of the same locus and in case of dominance, and at a different or another locus in case of epistasis. Depending upon who is influencing whom, the dominant or the recessive, we have different representative ratios, which we are going to discuss step by step, one by one, and we would reach the conclusion that the different types of interactions exemplify different types of modification products, and obviously, the ratio also changes, starting from the interaction of supplementary genes, that is, if there is a recessive allele which is influencing its effect on the dominant ones, non-allelic ones, then the expected ratio would be 9:3:4. It would become 9:7 in case of complementary genes, which also we would like to justify why is it called as a duplicate recessive epistasis. There could be dominant epistasis where a dominant allele is exercising its effect. The ratio would now get modified into 12 versus 3:1, and sometimes both the dominant alleles exercise a similar role, and now the ratio becomes nearly absolute, which is 15:1. There could be an inhibitory factor, as I said in the beginning, of a metabolic pathway, which would not allow any of the other dominant genes to function, and in that case this interaction would result in 13:3 ratio. And finally, a very interesting aspect of polymeric factors. One would assume here that there is a lot of confusion between polymeric factors and polygenes or the quantitative inheritance. How do we sort that fog out? We would see at the appropriate time. Let us first begin with the recessive epistasis, because this happens to be the most common and the most visible effect as we see even in our households. Here we would find that there is one factor which is producing a visible effect, and the recessive alleles of one particular gene locus, say for example small a, can mask the phenotypic expression of another non-allelic capital, that is the dominant genes. So, that means when will this dominant gene express itself? Only when the other homozygous recessive alleles are not present. The most common example of recessive epistasis we see around us are the coat color in mice. If we ask a child what is the color of a mouse, he says a mouse color. He obviously means gray. We do not know whether he is talking of brown or he is talking of white, because white he could understand that it is sort of a albino, and many people also have the albino mice as their pets these days. But then we find genetically that the coat color of mice is governed by two dominant genes, capital C and capital A, and there is a situation where none of them are going to work for themselves. So, these are the black albino and the agouti mice. The grayish mouse is called as an agouti mouse, and it results as an interaction between these two genes. So, that means here you would find that if we take C as an example, the small c is present, then it has an effect on all the dominant alleles. So, that means this mouse has to be albino regardless of whether you have double dominant A or a heterozygous A. This is not the situation in case of capital C, because capital C is a allelic combination which is interacting with A to give rise to an agouti coloration. Let us see this more clearly in a checkerboard clause. Wherever capital C and capital A are there, that means this particular mouse has resulted, or its coat, the type of melanin pigment which has come is of a brownish nature. Sometimes we find that their ears and their nose, they have a different types of pigmentation and color of hair. And if there are two small c's on the other side, in this case a female, then it becomes an albino. We are not bothered whether it is small a along with it or a capital A, it is not going to function at all, because in the presence of small c, because that is exercising the epistatic effect. In the F1 generation, the combination would have at least one capital C and capital A, and therefore, the F1 generation, all the progeny are going to be of agouti nature. Now, to a common person, to a common viewer, it may appear to be simple dominance, but then this is not the case. We should remember that there are two dominant genes on one side, and in the F2 generation, as a result of the mating, we would find that the ratio has now been modified into 9:3:4. Let us try to explain this whole phenomenon. Actually, what happens is that if we take B as the gene for albinism, then small b suppresses the expression of capital A. So, that means small b in a combination is epistatic to capital A. So, it is a case of recessive epistasis, whereas the capital B itself is responsible for the production of black pigment, and wherever there is A and B, then it will be neither black or albino, it would be the agouti pattern. So, that means 9:4:3 is the dictum for recessive epistasis. How do we explain the mechanism of 9:3:4? We would say that these two genes A or B finally are going to result in an agouti pattern. So, that means gene B must be synthesizing certain enzymes which are responsible for converting the precursor molecule into melanin. And there are different types of melanin pigments. We will just discuss some aspects of genomics with it a little later. And once this black pigment is formed, if gene A in its capital form was present, it would be capable of synthesizing an enzyme which could modify this black pigment into agouti, maybe a different type of melanin. And if gene B is not present, then how would the black pigment be formed? And if the gene A is not present, then how would the agouti be formed? In other words, this mechanism would suffice as far as the explanation of 934 is concerned. Another very important example and a very common one which we find are the sweet looking Labradors. We have 3 very cute puppies, but with different colors: chocolate, white and jet black. Now, this coat color of Labrador retriever dogs again is a case of recessive epistasis. It is believed that there is another gene which is E in its recessive state, and this recessive E happens to be epistatic to B, that is the yellow coat. Before going to the cross, one would immediately assume that wherever small e is present, then you would find that the yellow coat has to be there, because it is epistatic to B, and B is for black coat and small b is for brown. So, wherever the small b e is present in a homozygous form, it has to be yellow. So, it is exercising epistatic control over capital B, and this is how we have the origin of black and brown and yellow puppies, perhaps in a same litter. This window on genomics gives us a very interesting aspect. It has been known that the 934 has perhaps originated as a result of a mutation in nature, and this is how the gene would have come. Very meticulous analysis of extremely well preserved woolly mammoths taken out from the snows of Siberia, so something like -70. Nothing could be more near life as far as the preservation excellence is concerned, and it was found that they are about 40 to 50,000 years old. These woolly mammoths, some of them are even redheads. So, that means this type of mutation would have taken place at that time also in mammoths, and there have been some fossil records of Neanderthals, our ancestors perhaps, let us say, on the same tree, and they also have red hair and different types of pigmentation on their skin. So, that means it is the same gene which is present in the Labrador retriever dogs, in us. This is a gene which is called as MCIR. So, this is a type of a melanin, and this is basically a melanocortin 1 receptor protein which is controlled by a gene. It is this particular gene which would have got mutated, and which results in the formation of different type of hair. Now, the focus on genomics suggests that there are so many genes which existed in the past, and many of them are present in us also. So, this is a case of human genetics. Maybe we will be more rational and we will be more justified in our treatment when we come to human genetics. How are these genes now distributed to different races of human beings, and the whites and the blacks and so on, and the melanin pigments. Of course, environment also has a very profound effect in this regard, and but the underlying factor here is that whatever we are discussing in Labradors is a recessive epistasis, and it was very much there in terms of Neanderthals as well as the woolly mammoths. The second aspect is of duplicate recessive epistasis. This is a child when he is introduced to genetics in class 12, perhaps looks at this particular diagram with complete disbelief and fascination that how can two parents with white flowers cross and their progeny is purple. Because a child could assume that red and white if crossed could give rise to pink. There could be some sort of dilution, even that was not correct, as we had discussed earlier in terms of incomplete dominance, that it was not blending. But this is absolutely undigestible that two white flowers parents are giving rise to a purple one. So, this is what we are going to discuss now, and this is exactly in sweet pea, Lathyrus odoratus. Here I would again like to remind you that Mendel had not worked on Lathyrus odoratus. He had worked on garden pea. Well, one could assume with the stretch of imagination, had he really worked on it, the results would have been very confusing, and perhaps the laws as they stand solidly would not have been that firm, I would guess. So, William Bateson was the one who observed this phenomenon, and he saw that plants, true breeding varieties with white flowers, gave rise to purple flowers. He was fascinated, where did this pigment come from? So, the rationale came in terms of the genetic explanation. There are two genes, capital C and capital P. The moment we say that there are two genes, obviously they are non-allelic and they have different loci, and they control the same color, which is the white color, the white pigment in the petals. The purple color in sweet peas dominates over white color. There must have been a recessive mutation in nature, and this would have taken place in either C or P, so that when capital C or capital P, that means they are homozygous, they would have given rise to white. So, once the capital C changes to small c and becomes homozygous, and likewise P, they become white. So, that means in nature at least one capital C and capital P, if they would come together in a progeny, they would now help in the synthesis of beta anthocyanin pigments, which would mean that you have mauve or purple or reddish flowers. So, the dictum is that they have to be together in a dominant form. As we see in this pictorial depiction, we have a white flower. Now, the white flower has a genotype of capital D, capital C and small p, small p. So, that means both of them are not there. So, there is nothing, the second one is not there to complement the first. And likewise, the plant which has small c and capital P also bears white flowers. In the F1 generation, the progeny had now combined one capital C and capital P, giving rise to a purple flower. And upon the selfing of the progeny or the members of the F1 generation, the F2 gives the modified ratio of 9 purple and 7 white. So, this is how the famous 9331 gets modified into the 9:7 cross. So, we can see in the Punnett square that wherever we have small c, we have the presence of white. Wherever we have small p, we have again the presence of white. Naturally, this jinx will be broken when there is one capital P or capital C together. They have to be heterozygous. In that case, they would become colored, that is the purple ones. If we were to just look at the possible mechanism, we would again find that there are two genes which must be releasing their enzymes. The white substrate would require an enzyme to give rise to a white product. This product would be a precursor, which naturally would be requiring an enzyme B to become its purple. I always given a very commonplace example to my students who do not understand it. I always say that you require a pistol and a bullet to fire a shot. If you have an empty pistol, it is useless, and likewise if you have a bullet in your hand, it is also useless. You can throw it like a pebble, but it would not be a shot. So, likewise, if the gene C is not present, then the enzyme A does not function, then the white product does not form, and then how would be the purple product formed? And if by chance the white product is formed as a result of the presence of enzyme A due to the presence of capital C, capital P is not present. So, that means now you have an empty pistil and you do not have the shot. So, in any case, the purple coloration would be produced only as a result of complementation of these two genes. So, this is how we say that they complement each other. So, that means this is a theoretical pathway which can convert a precursor to an intermediate, finally to a purple extract. This is one way to interpret the complementary genes. A child naturally asks, you are talking all the time of complementary genes and you are teaching this in the chapter of epistasis. Where is the epistasis? Who has stood upon what? Because here you are saying that capital C and capital P, they are combining and giving rise to a purple product. So, how do we explain this term, phenomenon in terms of epistasis? Actually, small a together is epistatic to the other dominant gene, that is capital B, and likewise the small b when together is epistatic to capital A. So, in our present example, if there is a small c, then capital P would not function, and wherever there is small p, capital C is not function. So, therefore, this would be justifiably called as duplicate recessive epistasis. So, it is a double form of epistasis. We find that there are some children who are born who are deaf as well as mute. To a common person, they will say, okay, maybe these two genes are linked. A little knowledge of genetics can also be dangerous, like all the other aspects. So, immediately it comes to a mind, okay, this is a case of linked genes and there is no crossing over and these two genes have come together. Actually, this is also a case of complementary genes, and the probability of this type of complementation, because both of them are dominant genes, deaf as well as mute, and still you see that me and you, both of them are not deaf and not mute, but so that means it is the recessive which prevails, which again brings us to our chapter first, that there is a difference between dominance and prevalence. It would depend on what is the frequency of that particular allele in a population, which would again take our one lecture in the form of population genetics, when we are talking about gene frequency and its prevalence in a particular population. So, that means both of them, if they are coming together, these two genes, then a child would be deaf as well as mute. Well, it could be a mute and not deaf, all right, but then this type of combination is also prevalent. So, this is also an example of complementary genes or duplicate recessive epistasis. Then comes the dominant epistasis. Here, of course, this is a simple one, because a dominant gene in its dominant allele form is going to be epistatic, say, on another gene, capital B. If it is stopping capital B, then of course the small b has no say. So, that means here you would find that the same genotypes would be present in case of A or anything else, and small b as well as the heterozygous one. Here the modification of the F2 ratio is 12:3:1. 12:3:1. The most common example which we find is the fruit color of summer squashes. Of course, it is good to talk of summer squashes in winters, because still they are used for their soups. So, we find that there are different colors of cucurbita pepo, and it is white, yellow and green. Incidentally, white happens to be the most prevalent one, but we do not see much. We see only green kaddu or yellowish in the market. The white is rare, but then it is the one which is really dominant, because the yellow is recessive to white and yellow is dominant to green. So, that means green is the lowest and yet it is the most prevalent in the market. So the gene which is responsible for white is capital W, and it is epistatic to capital Y. If we just look at the crosses of a white fruit of say a summer squash or a gourd, we would find that it has one W capital and one Y capital, but does not matter, because capital Y is epistatic to capital Y. So in the presence of at least one, even one capital W, the fruit has to be white, and we cross it with another white where we have a heterozygous condition, the same one, we would find that in the F1 all of them are still white. We could take another cross which I have shown sideways, and this is a cross between white and green, and then the progeny also would be white, because we only understand that if there has to be at least one capital W and capital Y, and at least one, even one capital W would mean that it has to be white. And in the F2 generation there are 12 whites and 3 yellows and 1 green. So here this is a case of dominant epistasis. The small w is not exercising any epistasis, it is the capital W. So it is a case of dominant epistasis. This would also mean that in the mechanism the white precursor has to be changed or modified into a yellow pigment by a gene C, for example, and that yellow pigment needs to change into a green pigment with another gene G, and if there is a W present, then neither C works nor G. So the white precursor remains white. But then this is a case of dominant epistasis. Again, this particular gene in terms of its frequency would be so low in a population that you would seldom see a white gourd or a white summer squash. One may even treat it that it is diseased. No, it is a case of gene interaction. And if we could explain the biochemical mechanism, again it would be the W allele which is in fact stopping the appearance of green as well as yellow in both the situations. Well, the dogs which we see around, if you see a litter of say 6 puppies, all of them look good and cute, but then all of them have different patterns, and this also would be and different colors. And this again is a case of dominant. We are not talking about Labradors, there the coat color was different, and here the brown dog, if it mates with a white dog, then you will have white, black and brown. So you needn't have a black father or a black mother to produce a black puppy. It could be the two, of course they would have the genes, all right, which would show dominant epistasis. One of the very classical examples which have been mentioned is in horses. Horses are seldom kept these days, but the people who have various stag forms, they observe throughout their life that the horse as it ages shows a progressive silvering or a lightening of its coat with age. So that means at the end it may even become whitish. So that means this genotype would have a different type of gene of epistasis. The ones which do not would not show dominant epistasis. So that means the progressive change in pigmentation of horses is also a good example of dominant epistasis. Here we talked about one gene, and what about if both the genes are dominant, like we just discussed a few minutes before for the duplicate recessive epistasis. This particular phenomenon of duplicate dominant epistasis was first discovered by Professor G.Shull. You remember G.Shull is associated with the famous term heterosis or hybrid vigor, and a mule, there can be no better example than a mule, which has the better qualities of a horse as well as a donkey. So he worked on a weedy plant which is shepherd's purse, Capsella, bursa pastoris. Now this particular plant has two types of capsules for the seeds. One is triangular and the other one is ovoid. And we find that the triangular capsules actually they would dominate. And there are two genes in their dominant form. If there is gene A, it also controls triangular shape, and surprisingly gene B, which is again non-allelic, also controls the triangular. So that means whether it is capital A alone or B alone or both of them together, it doesn't mean anything different. It means all the same. The capsules have to be triangular. When would the capsules be otherwise? Only when both of them are present in a recessive form. And we remember from our basic lesson of a checkerboard that this genotypic combination has to be only 1 out of 16. So that means we needn't even look at the checkerboard. We will simply derive the idea that the ratio has to be nothing else but 15:1, because there is just one such exception where we would find that both of them are in a double recessive plant. This is the plant of a shepherd's purse, which has the triangular seeds and the ovoid ones or the capsules. In dominant duplicate analysis, we would find that the cross between a triangular or an ovoid will only be, as I said before, double recessive or double homozygous recessive. We would find that the triangular capsule ones appear in the F1 generation and the ratio becomes 15:16. So just one at the end is small a, small b, small b. What would be the mechanism in this case? When we say that A is also responsible for triangular and B also, that means both of the genes release products which convert the products into a triangular capsule. And if none of the capital alleles are present, as we have shown below in small a, small b, small b, it's a simple case of blocking of both the pathways, because either of the pathways would have given rise to a triangular, but here none of them is working. So this is a case of duplicate dominant epistasis. Now, what are the other permutations which are left? Both recessive, both dominant, one dominant and one recessive. Now, the only thing which remains is one dominant and the other recessive. This is also what happens and forms a very interesting example. And this is an interaction of an inhibitory gene. Actually, what happens is that there are two pairs of dominant factors again controlling here. One of them inhibits the effect of the other. If you were to look, for example, an entire thousands of acres of paddy fields, you would seldom see any leaf which is purple, but actually the purple pigmentation is dominant over the green one, yet it does not appear because there is some inhibitor which is present. The gene capital P, which is for purple, is actually dominant over small p, which is green, and there is another factor capital I which is present in the rice plants, and this is called as the inhibitory factor. This would stop the production of the anthocyanin pigment which is controlled by capital P. So naturally, wherever capital I would be present, it's immaterial what the other genotype in terms of P is. It could even be capital B doesn't matter, or small p doesn't matter. So P capital is for purple, small p is for green, and I for inhibitory, and I is having an inhibitory effect of P. So that means if you are going to cross a purple and a green, and if the combination has at least one capital I, all the plants in the F1 have to be green, and when they are bred in the F2, the ratio becomes 13 green, 3 purple. So that means now this ratio becomes 13:3. This is an interesting aspect of the genes which are called as inhibitory interactions, because here at least one capital I would be needed. The same we find in case of different types of poultry, in chickens also, because there are different varieties, their plumage is different. You must have seen one desi chicken which is not white, which is brownish and reddish. So that means their plumage is also different, and it also shows a 13:3 interaction, which is of the the inhibitory factors, or we can in other terms call it as a duplicate, and if not duplicate, then a dominant recessive interaction. Finally, we have an interaction of polymeric factors. Before dwelling upon it, let me just remind you that we had talked in great details, including some of the numericals, of polygenic inheritance and of quantitative genetics, where, if you remember, I had tried to explain how a progeny or a son or a daughter of two average sized individuals could be much taller than them. And of course, we had wittingly talked about intelligence being controlled by 27 genes and so on, and I had even witted that all children think that they are always more intelligent than their parents. It may not always be the case, it could be vice versa. So that means in this case it was a numerical addition of the dominant genes which had a contribution to make to the basic character, whether it was our skin pigmentation of melanin, whether it was our height, whether it was our intelligence. They are all quantitative, and for that matter even our ability to solve mathematical problems, that is also a quantitative trait. So that means if we had 4 capital genes, then we would be more intense than the one who has 3. So here the term polymeric also gives us a faint idea that it has also something to do with quantitative genetics, because a number of genes are controlling the same character. You go to the market, you will find that there are 10 different colors of the wheat which are available, having starting from deep red to little red, then amber colored and whitish. To a person who's lost, the TV would always show that the person has a wheatish complexion. But to a geneticist, the question comes, which type of wheat? Whether it is reddish or pinkish or MP wheat, which one? So that means we find that this is a nearly quantitative character, and the hue or the type of color of the grain which we have in a wheat seed also depends upon an interaction. So that means if we take 2 genes into account, capital A and capital B, if they are together, that means one of A and one of capital B, then the phenotype would be most intense. If we have one of them, then it will be less, and if we have none of them, then we have a still different phenotype. So that means the ratio here would be of 3 phenotypes. Imagine we are talking of a dihybrid cross, and now we have a ratio which is which could even be 13:3. So that means only 2 types of phenotypes. We could have say 9:6:1. So that means 3 types. A fruit shape of summer squashes, not the color but the shape is a classical example. Even the eye color in Drosophila, in the common fruit fly, also there. Wheat color, that is the pericarp color of wheat, and pigs also have a different coat color. They also belong into the same category. Let's look at one specific example of the pericarp color. The 2 genes which are responsible are C1 and C2. Now you should remember very clearly that C1 and C2 have to be present together, and it cannot be that you say, okay, let's put 2 C1 together. So if you had put C1 both of them together, then it will become polygenic inheritance, because then we are not bothered which ones, then we are asking us a question, how many. Here we are saying how many and which ones. So that means the C1 or C2 alone would give rise to a light red pericarp, and the absence of both would give rise to just a pericarp which is the colorless. So this is how we see that these particular different types of variants of the summer squash take us away from the monastery and the botanical garden of Mendel, and there are other exceptions in the world also which are due to interactions.
Thank you very much, sir. Thank you very much for explaining the difference between the modifying genes, epistatic genes and polygenes. Definitely, we have always been confused by what are polygenes and how to differentiate it from the epistatic genes. Definitely, I would like to continue with this discussion with you across polygenes and epistatic genes. Dear students, if you have any queries and comments, please do catch up this lecture once we upload it on the YouTube. And thanks for watching. Thank you, sir, for coming to our studios and enlightening us on this.
I'm equally delighted.
Thank you, sir.